Hi, my name is Begum. In this presentation, I will explain how fibroblasts, a type of skin cell, can be turned into neurons through direct reprogramming. This process was pioneered here at Stanford University and was published in the scientific journal Nature in 2010. First, let's take a quick look at fibroblasts and neurons. Embryonic stem cells give rise to three major cell lineages, or germ layers. These are called endoderm, mesoderm, and ectoderm. Each cell lineage, in turn, gives rise to many different types of cells in the human body. Endoderm includes cell types, such as thyroid cells and pancreatic cells. Mesoderm includes cell types, such as red blood cells and fibroblasts, while ectoderm includes cell types, such as glial cells and neurons. In this presentation, we will be looking at how fibroblasts, a type of cell that stems from mesoderm, are directly reprogrammed into neurons, which stem from ectoderm and are unrelated to fibroblasts. This process demonstrates the first conversion between adult cell types from different germ layers. Cell reprogramming is defined as a switch in gene expression of one kind of cell to that of another unrelated cell type. In this research, 4 retroviral plasmids are used. Each of these 4 plasmids contains a different reprogramming gene, or as we learned in class, a transgene. The 4 transgenes needed in this process are ASCL1, BRAIN2, MIT1L, and RTTA. ASCL1, BRAIN2, and MIT1L are the neuronal reprogramming genes, while RTTA is a gene that is required for this particular system to work. In the remaining portion of this presentation, I will be using one of these transgenes, ASCL1, as an example. The other transgenes also undergo the same procedure. The retroviral plasmids that contain our gene of interest, in this case ASCL1, along with capsid packages and replication proteins, which altogether enable the viruses to form and proliferate, are put into human embryonic kidney cells. Here the proteins and RNA package into a transmissible form and replicate. After 2 days, these viruses can be harvested from the kidney cells. Let's take a closer look at a tissue culture dish that contains these human embryonic kidney cells. As viruses form and multiply in a given cell, they leave the cell and migrate to the liquid medium. Once most viruses migrate to the liquid medium, the liquid is removed from the tissue culture dish and filtered to remove any kidney cells that may be in the medium. Now we have a medium full of viruses carrying the ASCL1 reprogramming transgene. This medium is added to a new tissue culture dish with fibroblasts. Once added onto fibroblasts, viruses enter the fibroblast nuclei. Here is a closer look at a fibroblast nucleus in the dish. The virus enters the nucleus and integrates its RNA into the host cell genome at a random locus. In order for a fibroblast to successfully turn into a neuron, all 4 plasmids carrying the different transgenes need to be integrated into the host cell genome. This is one reason why researchers use hundreds of thousands of fibroblasts in this process, since the probability of all 4 plasmids integrating into one fibroblast is relatively low. Next, we look at what happens once all 4 plasmids are integrated into a fibroblast nucleus. First, the RTTA gene, which was integrated into the host cell genome by its virus, is transcribed and ultimately translated to make RTTA protein. The RTTA protein and tetracycline molecules that have been added to the cell medium bind at the tetracycline inducible promoter to induce the transcription of the ASCL1 transgene. As a result of this process, ASCL1 protein is produced. ASCL1 protein then binds to promoters of neuronal genes in fibroblasts and activates transcription of various neuronal proteins, such as ion channels and neurotransmitter-producing enzymes. These proteins are abundant in neurons. Now, we have an overexpression of neuronal proteins in fibroblasts. As a result of this overexpression of neuronal proteins, fibroblasts begin to behave like neurons. After about 12 days, the fibroblasts have acquired a more neuronal shape and grown dendrites. Measurement of their electrophysiological properties shows that they also function as neurons. For example, they fire action potentials and form functional synapses. This conversion process has already been used to model neurological diseases from patient-specific fibroblasts, and in the future has the potential to be used for patient-specific transplants into people afflicted by degenerative diseases. Thank you for your attention.